So I think we left off right around here, looking at anatomy, and there's several things on here we'll go into in a little bit more detail. So we left off talking about the primary cell wall and the secondary cell wall. And so all plant cells have a primary cell wall. That's pretty flexible. That one, the number one with a degree symbol is how we denote primary, the word primary. And so all plant cells have that. It has mostly cellulose as its component, but we learned last time that there's other things in there like pectin and hemicellulose, glycoproteins. With the secondary cell wall, that is stronger than the primary cell wall. It's thicker than the primary cell wall. And one of the reasons that it's stronger is that it has an additional chemical called lignin, right, which is a branched polymer, which we don't have to get into the details of, but it is very strong. And as the artist drew this, you can see the plant cell, the secondary cell wall is much thicker in appearance compared to the primary cell wall. So that's one of the reasons, if you've ever driven by a paper making plant that makes paper and you smell those weird, nasty smells, it's chemicals, really abrasive, strong chemicals that are used to try to break down lignin and get a lot of that out before they make the final paper. So good quality paper doesn't have a lot of lignin in it. If you have too much lignin in paper, it gets kind of brown and brittle over time, it ages. But anyways, for the plant, it's awesome because it's super strong. Okay, another thing that we need to look at, going back to this image, is those, like, I call them tunnels between the cells. All right, so looking at, it makes sense that neighboring cells would have a way to like pass chemicals back and forth. And so those tunnels will do that. It's an open system. It's lined, so the cell membrane of one cell kind of lines the tunnel and with the neighboring cell. And so it's open. It's small though, like the nucleus can't get through, but other things, small chemicals or proteins can get through. So with that idea, we're looking at the plasmodesmata. You need to know that term. The plasmodesmata, those are the tunnels between neighboring cells that allows for communication. So you can have hormones going between cells, proteins, enzymes, other communication chemicals that are being used. And so here you can see a couple of images of it. The left one is an actual electron microscope picture, and the right one is an artist rendition. So neighboring cells need to have a way to communicate. So plasmodesmata will do that. And then if we start looking at a plant cell and pointing out some of the unique aspects of them compared to humans and other animals, of course, probably most of you can immediately come up with the idea that they have chloroplasts. And so we'll talk more about chloroplasts when we get into photosynthesis, but they're kind of funky. They have this outer membrane, and then they have an inner membrane that surrounds them. So they have actually this two membrane system. So I need you to know that there's an outer membrane and an inner membrane. So they call it the double membrane. And then they have an internal membrane. So that internal membrane, all that green that you see, that's like the internal membrane that's floating around inside the chloroplast. And that whole, like if I say, oh, look at that internal membrane, the other word for that is thylakoid. Okay, so when you consider the entire membrane, so I don't like the way the artist did this. They are pointing to like just this depth region. They should have like put a whole circle around that entire green internal membrane. All right, so that entire internal membrane is called the thylakoid. And then if you zoom in on just, you can see how some parts of the membrane are stacked, and then other parts look like bridges between the stacked regions. And so you have stacked regions and relatively unstacked regions. So those get different terms because when we get into photosynthesis, certain things happen in the stacked regions, some stuff happens in the unstacked regions. So the stacked regions, we call those the, come on. Oh, it's being stubborn. Let's see if I can write this small enough. GRANA, G-R-A-N-A, not really. G-R-A-N-A is the stacked regions. That's plural. If you talk about just one, it's U-M on the end, GRANUM. I usually say GRANA because we usually talk about plural. But, and then the relatively unstacked regions are called, unfortunately, the stroma lamellae. I'm going to put it on a separate thing so I can write bigger. So the unstacked regions are called stroma lamellae. I don't know why they decided to name it that. I would not have chosen that, but stroma lamellae are the relatively unstacked regions. So when we get into photosynthesis, you may hear those terms again. So I'm pointing those out now to kind of get that in your head, and then we'll come back to it. So that's where we see photosynthesis cooking along, chloroplast. So depending on what part of the plant body you're looking at, you might not see any chloroplast, right? So if you were a plant, where would you maybe not put your chloroplast as you're building your body? What would be like, yeah, we don't really need it here. What do you think? Roots, yeah, why bother? And so root tissue should really not have any chloroplasts unless they're aerial roots. So orchids, on the other hand, orchids that hang out on branches of other trees and they dangle their roots, they're green. The roots look green because they do have chloroplasts. But the typical plant that buries its roots in the soil, it doesn't make sense to have the chloroplasts in that tissue, right? So depending on what part of the plant body, you will see chloroplasts. In other parts, absolutely not. So we'll get more into how amazing chloroplasts are, but let's look at some other. So a plastid is like the umbrella term that we give, referring to organelles like the chloroplast. And so the chromoplast is another example of a plastid. And so it's like, what happens in a nutshell, what happens is the plant will make these plastids, and then what they see happening is some of the plastids start to like morph into, well, this is going to be a chloroplast. This one's going to be a chromoplast. So they all start out as plastids, and then with different gene expression patterns, they'll shift into these subcategories. And so chromoplasts are another example of a plastid. And they don't do photosynthesis, but they're colorful. They're not green. There's some other color. Yeah, I mean, you can think of it like that. They all start out as these plastids, and then there's this plasticity to it. You know, like they have different, what's the word I'm looking for? Not really destinations, but destinies. You know, they have different destinies of how they're going to end up when they're fully ready to go. So chromoplasts can be super important, even though they're not going to do photosynthesis for the plant. You could see that they're colored in lab yesterday. Did you look at some chromoplasts? I think tomato and maybe red bell pepper might look something like this. And so, yeah, this is a red pepper cell. And so, you know, for a plant, being able to have parts of your body colorful, that can be very, very important. And so chromoplasts can serve that function. There's other pigments out there that aren't necessarily in the chromoplast. So chromoplasts are chock full of these different pigments. But sometimes we see pigments showing up in the vacuole, which I'll talk a little bit more about. So it just depends, but these can be very useful. And then some other plastids are called leucoplasts. Leuco referring to colors because they don't really have any bright pigments inside. And so these are two subcategories of leucoplasts being shown here on this slide. Amyloplasts on the left and alloplasts on the right. Amyloplasts, you might see that purple tinge. It's because someone stained that plant tissue with iodine. And then what happens is it will stain starch purple. So anytime you see purple, those are amyloplasts are really, really good. If you look up the front half of that word, amylo means starch. And so it is storing starch. So white potatoes are famous for that. You'll look at that in lab eventually too. And then a lyle plants, the idea of an avocado, I believe you had one of those yesterday. And taking a little bit of avocado flesh and looking at under the microscope, you would see how those are lyoplasts. So now you know why avocados are super tasty, but you should eat them in moderation because they're chock full of lipid fats. So that's what the lyoplasts are doing. They're storing all those fats in the avocado tissue in this particular case. So those are some other examples of plastids, in particular leucoplasts. So we'll come back to certainly the chloroplast in more detail, and you'll see some more of, like you'll see amyloplasts, I believe, next week in lab. So ways to store important molecules for the plant. Mitochondria. So mitochondria, we have mitochondria, plants have mitochondria. Here you're seeing very typical, if you've taken any other biology class where they talk about mitochondria, that's the same idea. And so again, mitochondria have two membranes in plants, at least in the higher plants. You see that outer membrane. And then they have an inner membrane that gets all folded up like that. And so that folding of the inner membrane allows for great surface area. And so the mitochondria is super important for you as well as for plants because that's where they're making a lot of their ATP. And that's what we do with our mitochondria as well. So mitochondria are very important for the plant because they have to make their ATP just like we do, and they do it very similarly to how we do it. So that's a parallel to humans and other animals. And then I talked about this the other day, vacuoles. So this is especially important for plants because they don't have that skeletal system, as I mentioned the other day. So they don't have that nice internal skeletal system to keep them upright and fight gravity. They have to do other strategies like a secondary cell wall. That's a really good way to fight gravity and has good strength. The primary cell wall also contributes to that. But amazingly, the vacuole does too, especially if it's nice and filled up with water. So that central vacuole tends to be the biggest organelle. So you often hear, if you take a class where they talk about like animal cells, they say the nucleus is the biggest organelle. That's true in animal cells, but they forget in plant cells, it's actually the central vacuole. That's the biggest organelle. And really, if you think about it for a second, that makes sense because you want that central vacuole chock full of water so that it's exerting water pressure, sugar pressure against the cell membrane, which then pushes against the cell wall. So then you have a nice plump area of tissue, and that helps keep the leaves nice and flat so they can get good access to sunlight. It keeps everything upright so they're not flopping over on the soil. Okay, so the term given for the membrane that surrounds the vacuole is the tonoplast. So please get that one nailed down as well, the tonoplast. And so the tonoplast is mostly for water storage, but there's other things in there too, enzymes, pigments, proteins. If you've ever dropped an apple and a few minutes later, or maybe a half hour later, you notice there's a nice bruise now, a nice soft spot, what you've done accidentally is that area of the tissue, when the impact hit that part of the apple, a bunch of the vacuoles burst. And so some of the enzymes that are in there start acting on some of the cell components, start breaking down, and that's why it gets mushy like that. The bruised area of the apple gets mushy because the vacuoles broke open and the enzymes went to work. Okay, so central vacuole, very, very important. And when you look at them under the microscope, you might see something like this. So that top left picture, you're seeing the nucleus, and the bottom picture actually shows it quite nice too. So this bottom right picture is a scanning electron microscope picture, but you can see how all the organelles are pushed off to the perimeter of the cell. And then there's this big open white space. That's the vacuole. And so the vacuole is really hard to see unless you do a very finicky staining technique. So, but you can kind of indirectly tell if there's a vacuole there, why all that so-called empty space, that's where the vacuole is. Okay, so those are some of the internal organelles that I wanted to mention that we will be thinking about through the semester in more detail as we work through it. Let's kind of zoom out now. So we're done with our magic school bus and interior. Let's go and zoom out and start thinking about the overall body of the plant. So here, if you think about it, you know, how would you like to live your life in two different worlds? Their shoots and leaves are above ground, and then their roots are below ground. That's a very different lifestyle compared to us. And so typically when we study plants, we tend to think in those two realms because we've realized through a lot of research that what happens above ground can be very different to what's happening below ground. And I'll try to point out some examples of that when we get into plant growth. I'll try to show you how that happens. So, I mean, they're very different worlds. So things are going to work a little differently in some respects. But let's see what we got. Let's go above ground first. We have, of course, the stem tissue. And you can see that they have noted a couple of different spots of the stem tissue. So one, they're pointing to the node. The node is where you see leaves or branches coming off from the main stem. And you also see the so-called axillary bud. So we're going to talk more about the axillary bud. But that, in a nutshell, is a active area of growth during certain parts of the plant life cycle. Sometimes it's dormant and sometimes it's very active. So when the axillary bud gets the signal to go ahead and get active, it's going to either make a new leaf or a new branch. And right now, like the trees around here, the axillary buds are very quiet. They're dormant because it doesn't make sense to grow in the middle of winter. So the node region is there, they say that's the part of the stem where you see that nice axillary bud and you see branches or leaves coming off. And then the internode is just that area of the stem that doesn't have that stuff. All right, so of course you have leaves to think about in branches. And then right at the tip of the shoot, you see a shoot apex. So you need to know that. The shoot apex is another really critical area of the plant because it has, again, very active cell growth going on there. So I'll talk a little bit more about that. And then if you start to dissect the plant body carefully, you will see that there's going to be some vascular tissue. So we'll talk about that as well. We often use the analogy of our blood vessels system is kind of analogous to the vascular tissue of the plant. So I'll get into that. And then the roots, certainly we have to look at, there's also going to be root tips that we need to think about. So we talk about an apex, and whenever you see that, that usually means a tip of the plant body. So the root apex, the shoot apex, very important. Root hairs, we're going to talk about those as well. All right, so let's get into some of this. So these meristems, the shoot apex, the root apex, another term for that area is called the shoot apical meristem or the root apical meristem. And so that's being shown here on the image. Those are super important because it's allowing for active cell division. So there's very active cell division when a plant is not dealing with winter. They're going to be busy growing and growing and growing in their shoot apex and root apex. We also see that there's meristematic tissue, they call it lateral meristems. That's also active areas of cell growth. So if you take a stem, the plant stem, and cut it in a cross section, which is what they've done here, and you see that red ring of tissue in the stem, that is what we refer to as lateral meristematic tissue. So we need that to happen in certain plants. So if you think about the giant sequoias or the giant redwoods, if they're going to get a stem diameter that is so impressively huge, they need lateral meristems to do that. And so we'll talk about lateral meristems when we think about especially stems of big trees. So let me just show you an image of that to hopefully make a little bit more sense. So here you're seeing kind of the yearly growth here of a tree. And so you start out, very young, you got bottom right, and then as you get more years of growth, you see that the tree trunk is growing bigger in diameter. And so the lateral meristems, if you think about, maple trees and aspen trees, all of that, if you cut their stem and look at the cross section, it would look something like this. And we're going to talk about secondary tissues. So anytime you think about lateral meristems, that's when we start saying they're producing secondary tissues, which just means it happens later in their life. So initially, when they're a tiny seedling, all the primary growth is going on. They're getting longer and shoot and roots, but they don't really get into growing wider until they're a little bit more mature. So then we tend to say those are secondary tissues. They come later, okay? And so the lateral meristem, unfortunately, they have two words for it called the vascular cambium, which means basically the same thing as lateral meristem. Okay, so the vascular cambium is, I don't know if it's going to show up with, because my thing is red, but let's look at this biggest tree trunk, and you can see the like the dark pink area, and then you see all of this kind of purpley area. That boundary in between those two, that's the vascular cambium, right? So the boundary between those two is that it is a lateral meristem, and I'll talk a little bit more about that. Oh, and then there's another lateral meristem called the cork cambium. And the cork cambium is actually, when you start looking at the bark, it's kind of right there. The cork cambium is in the bark tissue. And so we'll get to that a little bit more too as we get into stem tissue. And so the cork cambium is important for trees because it's really helping to build a nice bark around the outside of the tree trunk, which trees need in order to stay physically intact and protect themselves. So lateral meristem, the vascular cambium is an idea, a cork cambium is another idea of a lateral meristem. And then you have this meristematic tissue that's called intercalary. Grasses do that. So that's why you have to mow your grass all the time. You can blame the intercalary. And so it's a area on the leaf blade close to the ground. So unfortunately, you don't zip it off when you mow. You don't reach it. It's low enough where your mower blades don't grab it. And so when you cut your grass, and then those intercalary meristems go, oh, I'm gonna grow again, and they just produce more leaf tissue. All right. So grasses are famous for doing that. By the way, when you mow your lawn, if you can get past the exhaust fumes of your lawnmower and you smell that lovely cut grass smell, usually most people like that. That's actually the plant more or less screaming that it's been injured. So it's their chemical way of signaling out to other plants nearby, I'm getting attacked. They don't really know well, actually they do know the difference, that it's a leaf blower made, the blades on the leaf mower versus an insect, for example. But that lovely smell is really them calling out and saying, I'm getting wounded, you might be next. So kind of sad to think about. Hence, I don't mow my lawn very much. All right, so now if we just look at the whole plant body and someone says, so what kind of tissue makes up most of the plant body? This would win, parenchyma. All right, so if you look at the whole plant body, parenchyma tissue is everywhere, everywhere. Every part of the plant body you can find it in. And so super abundant. It's made up just of parenchyma cells, which is what you're seeing here, a bunch of parenchyma cells all, neighboring each other. And you can see that they kind of are many-sided, but there's different sizes, and they're living cells, of course. So that means, do they have a secondary cell wall if they're living? No, they do not. All right, so they have primary cell walls. Their cell walls are just primary. They're flexible and thin. And that's what they would look like under the light microscope. And so, but they're all over the place in the plant body, both in the shoots and in the roots. By the way, the notes section of my PowerPoint slides, if you click on that when you're in my PowerPoint, you'll see that I have extra notes written in there so that if you maybe as you're in here with me, you can look to see what I have in there so that you don't reinvent the wheel and have to rewrite everything. You know what I mean? Like it's there. And so just look at your notes section as you go along. It has extra information. Here's some tissue, parenchyma tissue. That is also very important. So I think next week you'll look at parenchyma tissue under the microscope. So that's what you're seeing here. And I tend to say, yeah, parenchyma tissue looks funny under the microscope. And it's got this weird irregularities, thicknesses, and, around it. That image in the lower left there is showing a stem tissue of probably the best example I can give you of your experience with parenchyma tissue is if you've ever bothered to eat celery stalks, and you bite into celery and you don't always, sometimes there's like those strings that your teeth don't cut through all the way. That's parenchyma. It's so strong that sometimes your teeth have a hard time making a clean cut through it. So that's why sometimes the cut piece will kind of be stringy. It's because of all that parenchyma. And you can see how the parenchyma is grouped. All those cells are just tightly grouped together. All right, so parenchyma is very good. They're living cells, all right? They're alive, so they don't have a secondary cell wall, but they're thickened up. Their primary cell wall is thickened up a little bit, beefed up a little. And so you tend to find parenchyma around the outside perimeter, near the outside perimeter of stem tissue, like celery. And it's a good strengthening for stalks and stems. It's a good way to give a little extra physical strength. Okay, so the cell walls are thick and uneven, but they're living cells. So this is an option for the plant. They're like, well, I want some living cells there, but I need a little extra oomph of physical structure. And so they'll lay down some parenchyma in those for that. So very strong, even though they're living cells and only have primary cell walls. Okay. So the meristematic tissue is busy doing all of this. So we were mentioning the idea of stem cells. You can think of meristematic tissue as stem cells. And so the shoot tip, the root tip has that, the meristematic cells or tissues, and they're busy growing and dividing, growing and dividing. And then the cells that result from that have different destinies. Like some will become parenchyma, some will become parenchyma, and so on and so on. And so you can think of the meristematic regions as like stem cells, analogous to what you see in animals and humans. So here's another kind of tissue, sclerenchyma, and I did not name these. They have to give us such big, long names in science. It just seems like they love doing that. So sclerenchyma tissue, what we see are that there are a couple of different kinds. They're both composed of thick secondary cell walls, and so they're going to be dead at maturity. Once they get to their full size, they lay down that secondary cell wall, then the cell dies. And you can see, where's my, okay. And so again, because they're so structurally strong, they help with support. That's their main job. They're dead, so they don't do anything metabolic. It's just structural support. And so you're seeing two images here of what we call sclerites. So that's a subcategory. So you have your sclerenchyma cells, and then one example of that are sclerites. And you can see they kind of look funky under the microscope. You have these weird striations, weird like starburst patterns. So there's really no one way that sclerites look. They can have different visual appearances, but they're all thick, tough, with secondary cell walls, dead at maturity. And their sclerites tend to be scattered in different parts of the plant body. Your experience with spheroids, the best one I can give you is if you've eaten into a pear, which I hope you have, bite into a pear and you feel that gritty against your teeth, you're like, oh, that's kind of gritty. Those are sclerites. You're eating a ton of sclerites, all right, when you bite into the flesh of pears. And so they define them as their cells that are as long as they are wide. That's how they define them. But you can see there's variation in their pattern and how they look under the microscope. Okay, so you have sclerenchyma cells. Spheroids are one example, good for support. The next one, the other subcategory of sclerenchyma are fibers. Okay, fibers. They're very different. They're very long and narrow. Right? Pretty much any one you look at, they're long and narrow. And so on the right image, you're seeing a longitudinal, like someone just came along and shaved down the length of a fiber. So you can see they're very long. And then the picture on the left, what they did there is they took a cluster of fibers and cut them in cross section. And now you're looking at the cut end. And you can see all of that red is secondary cell wall. And then the little clear dot, that's just the internal empty cell. All right, so these fibers are super beefed up with that secondary cell wall, very, very thick. They cluster together. So similar to the parenchyma, but remember, parenchyma are living cells. Here we have clusters of fibers, but they're dead. But even though they're dead, they're super important. And I'll show you when we start getting into vascular tissue, you're going to see a bunch of fibers near there because they help protect the very precious vascular tissue. By the way, the cell inside that's all empty, they often refer to that as a lumen. It's just like an internal empty cavity. You have lumens in you. If someone were to cut open your intestine and look inside, they say, oh, look at the lumen. All right, so we have lumens too. It's not unique to plants. It's a word they give. Okay, so that was all examples of what they call simple tissue because it was just made up of one cell. You know, parenchyma tissue is just parenchyma cells, and parenchyma tissue is just parenchyma cells. Now we're going to get into complex, which just means there's more than one cell type that makes up that tissue. And so if we get into vascular tissue, that is an example of a complex tissue. I'm going to really zoom into two aspects of vascular tissue, the xylem and the phloem. So xylem, spelled X-Y-L-E, xylem. All right, so if you think about it, plants are multicellular. They're composed of millions of cells. You have to have a way to get things transported through the plant body, just like with us. We're super complex and composed of millions and millions of cells. So we have to have a blood vessel system that's going to deliver oxygen to our cells, take away CO2 and deliver hormones and deliver other chemicals. And it's kind of interesting that the artist even drew the example of the vascular tissue. Kind of reminds me of what you see in a human anatomy, where the veins, you know, they usually color code veins in a human, blue and red, depending on if they're delivering oxygen or taking away CO2. Anyways, so it's a good analogy. It serves a good analogy. And so we're going to look at vascular tissue. There's two types, xylem and phloem. So you need to know that those are two examples of vascular tissue. So xylem, super important. If we zoom into the xylem and we say, what does it look like? What's it composed of? We're going to see that it depends on the plant. Some plants have what we call the xylem type of vessels. All right, so this is a subcategory. Vessels are open at both ends, they're long, and they have a pretty good diameter compared to the other subcategories. So these are a couple of different examples of vessels. And you can see that they're longer than they are wide. And then you see all these perforations. So those are pores. So you have to imagine there's a bunch of vessels all clustered together and they're touching each other. And then those pores line up so that you can have exchange of material from one neighboring vessel to another. Kind of like the plasmodesmata, but a little different strategy here. So they're dead at maturity, which means they have a thick secondary cell wall. And all that kind of spiral that you see in the lower left picture, that's just showing how they're trying to show, they stained it to show the secondary cell wall is really designed in a way to create really good structural rigidity and strength. Now, look at, especially this middle, this stuff. It's quite wide and diameter. Let's look at the other type of xylem. The other type of xylem is called trachid. Okay, so vessels and tracheids. Tracheids are narrower than what we saw with vessels, and they tend to be tapered at the ends. They're also dead at maturity, though, but they have a very different look to them. They're always more slender than vessels. And then if you think about big trees that have really big tree trunks, really wide things, we also then start talking about rays. So rays are part of the vascular tissue that allow to just move substances like out laterally. So like that huge giant sequoia stem tissue can all get its necessary nutrients and water. Okay, so rays are just kind of helping with that flow across the tree trunk in this case. So they're living cells. They're parenchyma cells, but they have that striation head. They're all parenchyma cells lined up next to each other, and they're just helping with transport. In this case, with the xylem, we're going to see that xylem is really, really good. Let me just, yeah. So xylem is really good at transporting water. So if you're a typical plant that has put your roots into the soil, it's the soil water that the plant is tapping into. And so the roots will take up the water from the soil and it lands into the xylem tissue in the plant. And then that's like the internal highway system. Once the water gets dumped into the xylem tissue, then it can start coursing through the plant body and hydrate. So vessels, by the way, those you see in the more advanced plants. So angiosperms, which I often just abbreviate down to angios, A-N-G-I-O-S. Angiosperms have vessels. Wider diameter vessels like that are really, really efficient. And that's one of the reasons why they say angiosperms are so successful. There's more angiosperms on the face of the earth in this current age of Earth than any other plant. And they point to vessels as one good reason why they are so successful. It allows for much better and efficient water flow through the plant body. So angiosperms have vessels. Tracheids, on the other hand, they're also transporting water, similar to the idea with vessels, but they aren't quite as efficient. Now, angiosperms also have tracheids, so they have this ability to decide, so to speak, whether or not they're going to put down a tracheid or a vessel. And they can be quite savvy about it. So I'll explain more when we get into water movement, but at some parts of the growing season, they will say, I'm putting down vessels. That's my best bet. And then other parts of the season, they'll go, I think I'm going to mix in some tracheids here. And so angiosperms have the ability to have this flexibility. I can do vessels, I can do tracheids. And so you'll see both types in the angiosperms. However, if you were to peer inside a gymnosperm, a conifer tree, red pine tree, white pine tree, they're not quite as sophisticated with regard to their vascular tissue. Gymnosperms only have tracheids, all right? They're limited. So gymnosperms, we say that they're more ancient, I guess. They're not as advanced evolutionarily as angiosperms. And gymnos came first, and they're a little bit more primitive. And they figured out how to do tracheids, which is good, but that's all they could do. They can't do vessels. No, they're trying to get better access to water. So in the spring, when spring finally arrives here, which most of you will gone off to the end, we often have snow here by the time graduation happens. But anyways, imagine it's spring and we get lots of rainfall. Then the plant is like, ooh, there's lots of water in the soil. I'm going to lay down vessels, because that allows really rapid water movement. So it's a good kickstart to the growth in the spring. And so they get off to a running start, grow really vigorous, and it's super efficient to have those vessels doing that water movement. Then, June, July, they might hit some droughts. One downside of vessels is that if it's a drought period, it can cause what is analogous to us in embolisms in our veins. You get air bubbles forming in vessels under drought conditions, and that can be not good for the plant. It won't kill them like embolisms kill us, but it's not good for them. And so then they shift to making tracheids because embolisms are much harder to form. Yeah. Yeah. Yeah, they tend to shift over to tracheids. Yeah. And then they can go back to, because the vessels are permanent, tracheids are permanent. So they can divert, they can divert flow of water where they want. So yeah, they're pretty good that way. So xylem tissue, tracheids and vessels, both transporting water. By the way, the other idea with, where's my water word? The other idea with water, what's dissolved in the water is very important. And we'll talk about that. Minerals. So it's not just water that the plant is taking up from the soil. They're also accessing minerals. We tend to take a multivitamin for minerals, and hopefully the better option would actually be to eat a good diet that has minerals. But some people, think about a multivitamin as a supplement for minerals. Well, plants can't do that. They have to get their minerals from the soil. And so the minerals dissolve in the water, they take all of that up, and then they get that going through their whole plant body through the xylem. Okay, so vascular tissue, xylem, super, super important. The other type of vascular tissue is called phloem. Phloem. And this is, what you're seeing here are part of the phloem tissue, this term, sieve tube member. I want you to remember that. Now, depending on who is labeling the picture or writing the words, sometimes it's called sieve tube elements. Sieve tube elements. And like in Plant Phys, when I teach Plant Phys, that textbook author tends to use elements, I think. So I tend to do that. So if you hear me, sieve tube elements, sieve tube members, it's the same thing, right? So this is what we zoom into when we're thinking about what's the job of the phloem. It's the sieve tube members that will be transporting primarily sugars through the plant body. So xylem is for water and minerals. Phloem is sugars is a big one that's getting transported. And so now xylem, we said, was dead at maturity. It's just like a hollow tube system, which is very efficient, by the way, for water movement. That's really good. Yeah, phloem, they are not dead at maturity. They have a primary cell wall. They don't have a secondary cell wall, so they're still living cells. So that means they have cytoplasm. They're modified, though. When we look inside a sieve tube member, there's certain organelles missing. They're just not there. Which, when you start thinking about it, it's like, well, that kind of makes sense because you want a pretty good flow of sugar. You don't want sugar bumping into a bunch of chloroplasts or mitochondria as it's trying to flow through the entire plant body. And so they modify the Siv2 members, and then what they do is they put companion cells next to each Siv2 member. So what the authors or the artist is trying to show here with the yellow, those are the companion cells, and the tannish colors are the Siv2. So the companion cells are companions, one-to-one relationship. For every Siv2 member, there's a companion cell right next to it. And there's a bunch of plasmodesmata linking the two. Yeah, so there's all those tunnels. And basically, it's the companion cell's job to metabolically keep the Siv2 member alive. Because the Siv2 member doesn't have like mitochondria and certain other organelles. And so it needs some help metabolically. And so we see the companion cells helping out with that. And then the artist also drew here these Siv plates. So these are just where the Siv2 members link up with each other. So it's just one Siv2 member on top of another one, on top of another one, and they link up with those Siv plates. And it's all open. Okay, so those look like a sieve or pores, but it's all open, like membrane or anything blocking that. It's just an open system. All right. And so now you have to imagine sugar and water, sugar dissolved in water flowing through those Siv2 members. Okay. And it has to go throughout the whole plant body. So mostly when we think about photosynthesis, we think about plant leaves, busy doing photosynthesis, making all that sugar, but then they got to get it to the rest of the plant body. Okay. Roots generally don't do photosynthesis, so they're lacking sugar. And so the Siv2 members will be responsible for getting that sugar.
So the companion cells, are they found like elsewhere in cells, or is it like
their sole function? They're dedicated to helping Siv2 members. And then I said here they're found in more Siv2 members. They're found in more advanced plants, which are the angiosperms. Gymnosperms don't have Siv2 members. They have what we call Siv cells. So it's not the same thing, but it's the similar function, but they're a little physically different. Actually, truthfully, we don't know a whole lot about Siv cells. Almost all the research is done on Siv2 members because, of course, we care more about angiosperms as humans than we do the gymnosperms because we eat so much of the angiosperms. So Siv cells are still kind of mysterious, and we see those in more primitive plants like the gymnosperms, and then we'll find out that there's other plant groups that have vascular tissue, but again, they'll just have the Siv cells. They don't. Siv2 members are much more advanced and sophisticated, and along with those, companion cells. So, okay, we'll stop there and finish up the tissues, and then I posted in the next slide set on plant growth. So it's there if you want to take a peek at that before Monday. We'll get into that on Monday, I'm sure. So try to get your iClicker up and running, get it registered, and then click on my class and see if you can get that squared away. We'll try to do iClicker next week.